Hello everyone, and thank you for attending my poster presentation. My name is Paula Tatiana. Today, I'll be presenting our research titled Disruptive Effect of an Antimicrobial Peptide from Acropora digitifera on the structure of the AcrAB-TolC efflux pump of E. coli. This research was conducted in collaboration with Juan Rodrigo Salazar and Jorge William Arboleda. In recent years, bacterial antibiotic resistance has become a significant global health challenge. In 2019 alone, it contributed to around 1.27 million deaths worldwide, with nearly half of these cases caused by gram-negative bacteria. One of the main mechanisms behind the resistance is the use of efflux proteins. In gram-negative bacteria, efflux proteins from the RND family form a tripartite structure that actively transports harmful molecules, such as antibiotics, out of the cell. In this context, antimicrobial peptides, particularly those derived from marine organisms, show great potential as broad-spectrum, stable, and low-toxicity agents. So our study focuses on antimicrobial peptides from the coral Acropora digitifera, assessing their potential to inhibit the AcrAB-TolC efflux protein in E. coli. We start by identifying a 48 amino acid antimicrobial peptide from the transcriptome of the coral Acropora digitifera. We then generated 5 fragments of this peptide to analyze their properties. We use molecular docking to assess binding affinity between the peptide and the efflux pump. We also perform 100 nanoseconds molecular dynamic simulations to evaluate the structural stability of these peptide pump complexes. In a previous stage of 48 amino acid antimicrobial peptide was identified in the transcriptome of the Acropora digitifera blastula stage. From the full-length peptide, 5 fragments were generated, and their structure and physiochemical properties were characterized. The docking results showed that the full-length peptide exhibited a binding energy of minus 140.04, while a 15 amino acid fragment demonstrated the highest binding affinity. During the simulations, we observed structural changes in the efflux pump when bound to the full-length peptide at around 90 nanoseconds. For the 50 amino acid fragment, the disruption occurred around 60 nanoseconds, causing a rupture in the TolC channel structure. These observations suggest a potential mechanism through which the peptides interfere with the pump's function. These findings are significant because the RND type efflux pumps are essential for drug resistance in gram-negative bacteria. Our results indicate that the peptides from Acropora digitifera may effectively disrupt these pumps, thus providing a new avenue for antibacterial therapy. In conclusion, our research demonstrates that antimicrobial peptides derived from the marine organisms like Acropora digitifera could play an important role in combating antibiotic resistance. We believe this study contributes valuable insights into potential treatments against multi-drug resistance bacteria. Thank you very much for your attention.